Good afternoon, and welcome to Plenary 1. Please welcome Dr. Michael Boyle, President and CEO of the Cystic Fibrosis Foundation.
Well, good afternoon, everyone. It is great to be together. Welcome to the 38th North American Cystic Fibrosis Conference. It is really good to be together, isn't it? There's nothing more energizing than the world's CF community coming together to continue our all-out attack on this disease. And that means all of us, right? All of us gathered here in Boston, as well as the many people with CF, their loved ones, and so many other CF community members who are joining us virtually online for this conference. We have nearly 5,000 people from around the world, 42 countries represented, and many, many more online. Of course, we're all looking forward. I know we're all looking forward to diving into the science and the care topics that are the focus of the 259 sessions at this meeting. But first, let's take a moment to gather ourselves and focus for the task at hand. And to guide us, I have 3 words for us for the conference. And the first one is thank you. Thank you. Thank you for the part that you're doing in forming what people throughout medicine consider to be one of the most amazing medical mission-focused communities in the world. And that's the cystic fibrosis community. Thank you for your work. Thank you for your devotion. Thank you for your passion year after year after year for defeating the disease and all the challenges that come with it. So whether you're a researcher, a clinician, a CF volunteer, or especially a person with CF or a CF loved one, thank you. I hope that you see the impact that you've had and are having as we continue this remarkable story of progress. My second word for the conference is transformation. Transformation. What we have the privilege of being part of is the transformation of a disease, and even more importantly, transformation of the lives of people with CF everywhere. I know you can see that and you can feel it. Yes, we have much work to do ahead of us, but make no mistake that we are part of an absolute transformation that is creating a future full of hope. What was once a pediatric disease with short survival has been turned into a disease with adults greatly outnumbering children, and with median predicted survival continually increasing year after year. For people with CF in the US, it's now up to 61 years of age. When we step back, you have to think this transformation and how fast it's occurring is simply astounding, right? So let's talk about some of these areas of transformation as well as the work ahead of us to ensure that all people with CF, no matter what mutations they have, no matter where they live, will be able to experience this transformation. So at the CF Foundation, we are laser focused on making sure that we support the transformation of life with CF today, while also helping lay the groundwork for the continued transformation and cure of the disease in the future. For starters in this conference, you're going to see many stories of transformation regarding CFTR modulators, right? This includes numerous sessions on the impact of ETI on all aspects of life with CF. But also look for those sessions that provide a glimpse of what's ahead for the next generation of CFTR modulators, including Vertex's vanzacaftor, the once-a-day CFTR modulator currently under review by the FDA, likely to be approved as we head into 2025, as well as the potentially powerful modulators that are being developed by Sionna Therapeutics. Also pay attention to the growing evidence of what early treatment with CFTR modulators can do, completely preventing many of the symptoms and complications of cystic fibrosis. Now that's absolutely transformative, right? Then in today's plenary, we'll be hearing about the genetic technology that the CF Foundation is supporting to develop transformative therapy that will benefit all people with CF, no matter what CF mutations they have. Over the last few years, these genetic technologies involving both RNA and DNA therapies have moved past testing in the laboratory to now being tested in 6 different early phase clinical trials. Additional work is also ongoing in gene editing, which is still in the lab, but continues to progress and no doubt will eventually form the basis of a cure for cystic fibrosis. To advance all of this critical research, the Foundation is working to create an ecosystem that allows partners to develop new therapies as effectively as possible. You know about the Foundation's supported clinical trial network called the TDN, but did you know we also fund CF clinical trial networks around the world? And do you know about the CF Research Lab here in Boston, funded by the Foundation, that has a team of nearly 50 scientists pursuing genetic therapy research and working closely with companies to bolster and test their emerging strategies? And of course, along with the therapies of tomorrow, we're also committed to helping transform the approach to the challenges that people with CF continue to face today, whether that's infection, whether it's diabetes, which happens to be the topic of tomorrow's plenary, or reproductive health, which happens to be the topic of Saturday's plenary. Again, transformation in the cure and the care space. And then finally, what we're witnessing is a transformational moment, again, in the delivery of clinical care to people with CF, as reflected by the two CF Foundation clinical care position papers just published in the Journal of Cystic Fibrosis this week, and the many sessions at this conference focused on CF care of the future, including the one I just came from, which was totally packed. So we've talked about these first two words, these guiding words for the conference, thank you and transformation. The third word I want us to keep in mind, though, is urgency. 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 Urgency because there's a group of individuals with CF who cannot and will never benefit from CFTR modulators because of the type of CF they have. They're still experiencing every day the same horrendous, life-shortening CF that we've been fighting for decades. Urgency as well because we know they're members of the US CF community as well as around the world, and especially around the world, that don't have access to the care and medicines that they need that would transform their lives. This overall sense of urgency that we must feel really came home to me recently when I spoke with the parents of a young child with CF who's ineligible for modulators. After hearing my science update, they came up to me and said, "It's great to see all the things that are going on, but please, please convey to everyone you speak to the desperation that we feel, the urgency that we feel for our daughter, Gabriella." So let's make sure that as we dive into this conference, that we always hear in the back of our mind a clock ticking and have a sense of urgency for Gabriella and her family and so many others that are in this situation. So now before I end, I do have one other request. As we strive to advance research and improve care and address the many unmet needs of people with CF, it seems appropriate that the Foundation is starting now to develop our next 5-year strategic plan, which will launch in 2026. As we develop the plan, we're going to need to identify key priorities in cure, care, and community, and make critical choices about where to put our energy and our resources in the coming years. Identifying these key priorities will also help guide us in our financial planning so that we can be disciplined in our approach and ensure we maximize transformative approach to CF today while also maintaining over the long term what we need to do to be able to reach our ultimate goal, and that is curing cystic fibrosis. We want to make sure that this strategic plan that we go forward with reflects the broad needs of the CF community. That's why I'm asking all of you here and those of you online to complete a survey to inform our next strategic plan. For those in the room, you should look and you should find on your seat a card with a QR code for the survey. For those attending virtually, there's a link in the chat which will connect you to the survey. This survey only takes about 10 minutes. It'll explore the needs and the priorities related to research and care, education, and community programs. If you're here in Boston and you want to take some cards back to your colleagues and patients or families, stop by the CF Foundation's booth and pick up some extra cards and take it back with you. So I'm just going to say thank you in advance for your feedback to help inform our priorities as we look ahead to continuing to serve people with CF and their families. So as we head into this year's conference, remember these 3 words: thank you, transformation, urgency. Thank you, transformation, urgency. All of our work starts here this week at this conference, so let's get to work. Thank you. It's now my privilege to be able to to welcome the chair of the CF Foundation's Board of Trustees, Casey White. Casey has served on the CF Foundation's board for 19 years, became a board chair in 2022. Casey's the first person living with CF to serve as the board chair, and we're thrilled to benefit from her leadership in this new era of CF. She reminds us every day the why that drives all that we do. Please join me in welcoming Casey.
Thank you, Mike. It is an honor to stand before you, a dedicated group who plays such a pivotal role in advancing our mission. We just heard from Mike about his 3 words, gratitude, transformation, and urgency. And I would like to add one more, hope, a word that is deeply personal to me as a board chair, an assistant instructor of positive psychology, and as someone living with cystic fibrosis. In my studies, I've learned how psychological strengths like hope drive resilience, well-being, and progress in individuals, organizations, and communities. Over the past year, as I battled a multi-drug resistant bacteria, I realized that sometimes, even for someone who believes in hope as strongly as I do, it can feel elusive. But I've also learned that hope is always worth pursuing and is never truly out of reach. Hope is a powerful force that can drive change for people living with cystic fibrosis and for those committed to advancing care and research. First, let's define hope. Hope is more than wishful thinking. Instead, hope is active and goal-directed, a belief that despite challenges, a better future is possible and attainable through effort, innovation, and collaboration. I believe this is what brings you to NACFC. Second, hope is vital for people living with CF. In the CF community, studies have found that those with higher levels of hope have lower psychological distress, better adherence to treatment, and an overall greater quality of life, independent of disease severity. Third, hope is not only vital for people with CF, it is equally crucial for those who care for them, the clinicians and researchers in this room. Research indicates that healthcare professionals with higher levels of hope experience greater resilience, reduced burnout, and increased job satisfaction. Clinicians who maintain hopeful outlooks are better at fostering therapeutic alliances with their patients. In CF research, where we face complex challenges daily, hope drives us to take risks, ask bold questions, and persist through challenges, qualities essential for innovation and discovery. Consider the strides we have made in the last decade alone. The many advancements in care and research were made possible by a relentless belief that we could do better for people with CF, and we have. Yet our work is far from over. So what does this mean for us today? Hope is a practical tool that fuels our work, inspires our community, and unites us in our mission to transform lives. Before I close, I want to highlight one way to cultivate hope. Research shows that when we talk about hope, it grows stronger. By sharing what inspires us, we can deepen our own hope and spark it in those around us. So now I invite you to turn to the person next to you and share who or what gives you hope. And for those online, feel free to do the same in the chat. When you hear the chimes, please bring your attention back to the podium. Okay, let's begin. This is so... Wow, I apologize for interrupting these amazing conversations, but I hope you can feel the energy created by our hope. On the screen behind me are quotes from the CF community about where they find hope in their lives. As you can see, you play a significant role in generating hope in the CF community. Let's allow our individual and collective hope to continue to guide us in our important work. Thank you for all that you do for our community. I know that together we will reach our ultimate goal, a cure for all people with CF. Thank you.
High five, baby. Well, Casey, thank you for inspiring us once again. I was thinking about what gives me hope, and you know what gives me hope? When I look out at this group, you all do. If the amount of passion, talent, if this group can't get the job done, nobody's gonna get it done. So thank you for that hope. It's now my pleasure to be able to turn the program over to Dr. Stephen Rowe. Steve is the Executive Vice President and Chief Scientific Officer at the Cystic Fibrosis Foundation. Steve oversees our basic science, our academic research, our Path to a Cure programs, and also includes the CF Foundation's Therapeutics Lab and our biotech and pharmaceutical collaborations. Steve, as many of you know, has more than 2 decades of experience working in CF, is a pioneer in personalized medicine and translational medicine for CF. He joined our team in 2022 from the University of Alabama at Birmingham, where he cared for individuals with cystic fibrosis and served as professor and director of the CF Research Center. He still has an appointment as professor of medicine there today. Please join me in welcoming Steve.
Well, thank you, Mike, for that warm introduction, and it is great to feel the energy emanating from this audience, perhaps inspired by hope. And let me start by saying we are thrilled with the progress that we've experienced in the last year across all areas of CF, and look forward to working with you to build upon that momentum. Given the nature of CF as a systemic disease, we are working tirelessly to address CF from every angle, to unlock discoveries and developments across the spectrum of CF research. Along with and on behalf of the community of CF researchers in the room, from basic scientists to translational researchers and all those that work alongside them, we are currently supporting development programs focused on anti-infectives, CF manifestations, CFTR modulators, translational research, and the variety of aspects for genetic therapies. These programs span early discovery through early and late-stage clinical development. The breadth of this work together is intended to unlock the benefits for all people with CF and address a variety of consequences of the disease. Many of these programs are highly novel, pushing new frontiers. Thus, together, we are aiming to overcome scientific challenges by fostering collaborations in new ways. From those of us at the CF Foundation, you, the greater community of CF experts, and the clinical trial networks to give people with CF the best possible chance at realizing long and healthy lives. To that end, the CF Foundation currently has more than 35 programs in the drug development pipeline, including a potential new pancreatic enzyme, new CFTR modulators, anti-infective and anti-inflammatory programs, and genetic therapies, the last of which you will hear about during today's plenary. We have seen some exciting progress across the entire pipeline this past year. In addition to expanding modulators to younger age groups, efforts are underway to expand eligibility to modulators to people with CF caused by rare mutations. Further, the FDA is currently reviewing the application for approval for the once-daily triple combination modulator based on vanzacaftor for people with CF ages 6 and older with at least one F5 weight del mutation or other responsive mutation, potentially reaching more people with CF than has ever been reached by a modulator drug. And we have also seen steady advances in the development of therapeutics within the anti-infective arena, including novel approaches to target Pseudomonas. Despite this progress, it is still critical work to be done to help all people living with this disease. Approximately 10 to 15% of people with CF in the United States cannot benefit from CFTR modulators because they're not eligible or they can't tolerate them. Thus, it is more important than ever to develop transformative therapies for underlying cause of CF for these individuals, a task we approach with urgency. We also know success will open the door for curative therapy for all people with CF. Indeed, we are approaching the 5-year anniversary of the launch of the Path to a Cure. Our commitment to accelerate research to repair, restore the CFTR protein, as well as replace the defective CFTR gene. And as of this year, the CF Foundation has funded more than $500 million in research to advance transformative treatments for the underlying cause of CF. Through this initiative, we've learned a tremendous amount about the fundamental biology of CF, the challenges of genetic therapy to reverse the root cause of the disease, and conducted the evaluation of a number of novel therapeutic approaches. This progress, often marked by failures as much as successes, is a tribute to the enormous effort of the researchers here and beyond, pushing new frontiers into the science of gene delivery, gene editing, and mRNA biology, as we in the CF community seek to lead the way for this field to ultimately help cure a genetic disease of the lung and ultimately beyond. Over the last 5 years, we have seen important progress within academic research and discoveries, both within and outside of CF. This is where it all starts. The foundational groundwork is laid. I know that we would not have the benefit of CFTR modulators without understanding the basics of CFTR physiology and its cellular processing. And the same is true for progress in genetic therapies. Here we highlight just a few, from the cell biology to cell physiology and genetic science. These discoveries set the stage for the development of technologies that eventually lead to therapies for people with CF. So I want to take this moment to say thank you. Thank you to our research teams for your efforts, for bringing the best and the brightest together to tackle some of our most greatest scientific challenges. We are energized by your commitment, and we are committed to partnering with you as we keep pushing forward. And we are not just responding to the latest scientific advances, we are now driving research and positioning CF to take advantage of emerging developments in CRISPR and other gene editing technologies. Our past success and the ongoing support of the CF community give us the financial resources, expertise, and influence to help put individuals with the greatest need first in line for the next wave of innovation. This work has come to fruition, allowing the launch of an era of genetic therapy clinical trials in CF, including 8 currently operating. This includes clinical trials testing mRNA delivered by lipid nanoparticles, ASOs to target approachable mutations, and CFTR gene delivery with engineered viruses that have shown to be effective in other genetic diseases. It's important to note that not every attempt will be successful. But even clinical trials that don't result in new treatments advance our understanding and provide useful insight into how to improve future studies. And as we approach the 5-year milestone, our path to a cure is going strong. We have further developed an infrastructure to expedite research, including access to scientific experts and advice, lab and research tools and techniques, an extensive patient registry, and the TDN, a highly effective and dedicated clinical trial network. And we are fully committed to continue to invest aggressively into the most promising new pioneering science to lay the groundwork for genetic therapies, such as RNA therapy, gene delivery, and gene editing, and the means to advance them, as you'll hear about in the plenary. In summary, we remain focused on each of these components that we believe are a critical part of the CF research ecosystem necessary for success. And we are placing significant focus and efforts now in each of these areas to address current bottlenecks and to clear the path forward for these innovative therapies. And all of you today in the audience and those online are a critical part of our future success. Without you, there are no clinical trials to develop these transformative new therapies. So please join me in our commitment. We are all in this until it's done. And now I'd like to introduce 2 incredible individuals who are dedicated to advancing this work, Dr. Stan Siegwart and Joe Pilewski. Our first speaker, Daniel J. Siegwart, is a professor in the Department of Biomedical Engineering at the University of Texas Southwestern Medical Center. He holds the W. Ray Wallace Distinguished Chair in Molecular Oncology Research and serves as the Director of the Program in Genetic Drug Engineering, Director of the Drug Discovery Program in Biomedical Engineering, and co-leader of the Chemistry and Cancer Program in the National Cancer Institute designated Simmons Comprehensive Cancer Center. He was recently named as an emerging leader in health and medicine scholar by the National Academy of Medicine. His research laboratory utilizes materials chemistry to enable targeted nanoparticle delivery of genomic medicines. Their efforts led to an understanding of the essential physical and chemical properties of synthetic carriers required for therapeutic delivery. They developed selective organ targeting lipid nanoparticles, which was the first strategy for predictable tissue-specific mRNA delivery and gene editing, and ultimately aspire to utilize chemistry and engineering to make a beneficial impact on human health. Our second speaker, Joseph M. Pilewski, is a professor of medicine in the University of Pittsburgh and associate chief of her clinical affairs and pulmonary allergy, critical care, and sleep medicine division at the University of Pittsburgh Medical Center. In addition to patient care for adults with CF, his career has included basic and clinical investigations. His clinical investigations have focused on new therapies for CF, and as he's been the director of the CF Translational Studies Core and principal or associate investigator for over 20 investigator-initiated and multi-center clinical trials in CF. He's an active contributor to the CFTDN and serves as chair of the Clinical Executive Research Committee. As co-executive director of the CF Lung Transplant Initiative, he's contributing to research efforts to improve the care of individuals with CF before and after lung transplantation. So with those two outstanding speakers, I'd now like to welcome to our first speaker to the stage, Dr. Dan Siegwart.
Good afternoon, good evening. Thank you, Dr. Rowe, Dr. Boyle, and the Cystic Fibrosis Foundation for the opportunity to talk about the delivery of genetic therapies. Here are my conflict of interest disclosures, including Recode Therapeutics. I would like to begin by thanking key scientific contributors and colleagues who helped to conduct research and prepare this presentation. In addition, I would like to acknowledge Phil Thomas and Bill Skatch for introducing me to this amazing and supportive CF community. Let's start with the basics. Cystic fibrosis is a progressive genetic disease that affects the lungs, pancreas, and other organs. In people with CF, mutations in the cystic fibrosis transmembrane conductance regulator, CFTR gene, cause the protein to become dysfunctional. When the protein is not working correctly, it's unable to help move chloride, a component of salt, outside of the cell. Without the chloride to attract water to the cell surface, the mucus in various organs becomes thick and sticky. In the lungs, the mucus clogs the airways and traps germs like bacteria, leading to infections, inflammation, respiratory failure, and other complications. As someone who's lived in Texas for the last 13 years, I've come to appreciate the mechanical beauty of the automobile. It has a lot of interesting parts, especially the engine that can rotate thousands of times per minute. You can think about the CFTR protein as a car that moves chloride ions from one side of the bridge to the other, say from your home to your office. Now imagine that you get up in the morning to go to work, you walk outside and find that your car won't start. Well, a mechanic may be able to help fix the broken engine and help you get across that bridge to the office on the other side of town. That's like how small molecule therapy, modulator therapy, helps people with most CFTR mutations. But what if, when you go outside in the morning, your car is gone? That's like having a nonsense mutation in CFTR. There is no car, so modulator therapy cannot help. This is where genetic therapies may provide a path forward. Proteins like CFTR originally come from DNA in cells, which is like a blueprint to build the car. DNA is translated to messenger RNA, which is a copy of the instructions used in an assembly line to build proteins through a process called translation. Proteins are like cars. They do the work, such as move ions across channels to keep cells healthy. In the case of CF disease, This could be caused by a broken engine. What we'll talk about today contemplates fixing this problem at the root level, either through correcting the DNA instructions using gene editing, or providing a new copy of the instructions using messenger RNA. When a malfunctioning CFTR protein exists in this molecular setting, one can directly help CFTR function better through modulator therapy like Trikafta. The engine can be repaired by the mechanic. However, if that protein cannot be fixed, or if it does not exist at all, then we can consider genetic therapies that could replace CFTR protein or fix it permanently. mRNA would provide a new copy of the instructions to restore CFTR, and gene editing would correct the blueprint to fix or replace the gene. Now, we are entering into a new era of gene therapy, including gene editing approaches that are in early to late stages of clinical trials. Here's an example where delivery is less challenging. In an ongoing phase 3 clinical trial, Intellia Therapeutics is investigating a gene editing approach to eliminate mutant transthyretin that causes amyloidosis. Amazingly, following a single intravenous infusion of lipid nanoparticles that deliver CRISPR components to hepatocytes, the liver no longer makes this disease-causing protein. This is shown on the right, where the disease-causing TTR protein concentration is permanently reduced in the blood. Clinical trialists are seeing that this approach is working in the liver with long-lasting clinical benefit. However, CF is more challenging as a multi-organ disease, including the lungs. We also need to replace or repair the protein, not eliminate it. This requires more steps that we will discuss next. It is important to emphasize that genetic therapies are not the same as modulators. Trikafta consists of 3 small molecules. CFTR mRNA is more than 1,000 times bigger. Not only that, genetic therapies need to be delivered inside of cells. So it's challenging, involves multiple barriers to delivery, and the lungs will likely be the first tissue targeted for genetic medicine therapy in cystic fibrosis. So this genetic medicine approach clearly has potential, but how do we do this in the lungs? Well, let me take you back to about 6 years ago and tell you where we were. The Cystic Fibrosis Foundation and the National Institutes of Health assembled a group of scientific leaders to discuss a path to a cure, from basic biology and physiology to models and advanced delivery systems. At the end of the meeting, the conclusions were clear. We did not know which cells needed to be edited. We were not sure which genetic medicine platforms would work well for CF. We did not have models to be able to test new therapies, and the clinical roadmap was unpaved. So let's talk today about the progress that has been made. While nucleic acid therapies can treat CF at the DNA and mRNA levels, these molecules themselves can be degraded in the body. They need help to overcome delivery barriers. The helpers are synthetic and viral nanoparticles. To deliver genetic medicines to the lungs, local inhaled and systemic approaches may be viable. From the airway side, delivery nanoparticles must overcome the mucus as a key barrier to deliver genetic therapies inside of lung cells. Consider the top side of this cartoon as the view from inside the lungs. Alternatively, nanoparticles could be administered intravenously. Consider the bottom side of this cartoon as the view entering the lungs from blood vessels. From the vasculature, delivery nanoparticles must overcome the endothelial cell layer to access the therapeutically relevant cells that would translate to clinical benefit. Several approaches, including adeno-associated viruses, AAVs, other viruses and viral-like particles, lipid nanoparticles, known as LNPs, polymers, peptides, and other materials can form self-assembled nano-sized structures that encapsulate and protect genetic medicines and deliver them inside of cells. My area of expertise is in the chemistry and engineering of LNPs, which we'll talk more about today. Once delivery nanoparticles reach the lungs, they also have to deliver the genetic payloads inside of cells. This often involves cellular uptake through endocytosis, followed by endosomal escape as key challenges. Chemists and engineers design delivery nanoparticles, such as LNPs, to overcome these barriers and deliver mRNA to the cytoplasm, where the mRNA can be translated by lung cells into CFTR protein, or genome editors to the nucleus, where they can correct CFTR DNA. In both cases, providing the mRNA instructions or the DNA correction tools results in functional CFTR protein that can travel to the cell membrane and conduct chloride. Fortunately, as a key outcome of that 2018 meeting, the CF Foundation established the Path to the Cure, or PTAC. The CFF has since funded a variety of academic and industry projects aimed at tackling these difficult challenges, from delivery to editing to assays. Since I will not have time to discuss all of the important research going on, I encourage you to visit some of the exciting PTAC-related posters, workshops, and symposia noted here. Let's focus on CFTR messenger RNA therapy first. Similar to how the mRNA LNP COVID-19 vaccine delivered mRNA instructions for the SARS-CoV-2 spike protein to muscle cells to train the immune system, researchers are exploring strategies to deliver mRNA instructions to relevant lung cells to produce a correct CFTR protein. This approach would restore CFTR protein irrespective of the mutation. It will be a transient therapy that requires redosing. The person's mutation is still present in their DNA, but the cells use the new, correct instructions to make functional CFTR protein. Relevant cell types that express CFTR are now known to include epithelial cells, especially secretory cells, and ionocytes. So delivery systems will likely need to deliver mRNA to those cells for therapeutic benefit. In an example from Oregon State University, Gaurav Sahai and coworkers delivered CFTR mRNA through intranasal administration to mice that lack CFTR. Through electrophysiology measurements in the nose, they found that cells that previously did not make CFTR at all could now make the CFTR protein. Moreover, as shown on the right, the newly made CFTR protein from the mRNA instructions was functional and could conduct chloride. In ongoing clinical trials, CFTR mRNA encapsulated in lipid nanoparticles is being administered through a nebulizer. Oftentimes the same device is being used for other CF therapies. Prior to initiating animal and human studies, companies including Arcturus, Recode, and Vertex in collaboration with Moderna have utilized the human bronchial epithelial cell assay to measure functional restoration of CFTR. This is the same assay that was so valuable for prediction of clinical benefit of modulators. mRNA LNPs can be delivered basolaterally, or in the case of RCT-2100, as an aerosol mist to the airway side to better recapitulate the clinical setting. When this is done, and when LNPs are specifically selected and optimized to deliver mRNA to those important secretory cells, Recode has shown that this selective organ targeting SORT LNP mRNA therapy is dose-dependent, as shown on the left, in its ability to restore chloride transport in the HBE assay. The SORT LNPs used here are different than those used for liver and vaccine delivery, and were engineered for uptake into targeted lung cells. CFTR mRNA SORT LNP therapy can restore CFTR function as well or better than Trikafta in minimal function CFTR mutation HBEs, as shown on the right. In addition, RCT-2100 was administered into the lungs of CF ferrets harboring the G551D CFTR mutation. 2 doses of RCT-2100 was able to restore mucociliary clearance to the same level as 28 doses of VX-770. As shown on the right, using tracers and video recording to capture clearance of mucus from the lungs of CF ferrets, CFTR mRNA LNPs can significantly improve lung function. Seeing the mucus move up and out of the lungs is very encouraging for the clinical potential of mRNA therapies. We have talked about providing mRNA instructions to cells to make CFTR transiently. Let's talk next about genome correction strategies that could fix DNA permanently. Editing enzymes target, cut, and permanently repair the CFTR gene. If an edited cell dies, the edits are lost. However, if an edited cell divides, the new cells will contain the same edits. To achieve permanent, lasting CFTR restoration, the ideal editing target are the self-renewing basal stem cells that can differentiate into other cells in the lungs, including epithelial cells. Different editing strategies may have to be paired with different mutations unless the entire CFTR gene can be inserted. This approach could be permanent and require only one or a few doses. Still, delivery to basal stem cells may be required, which is a very difficult challenge. These cells, shown here in red, are not easily accessible to nanoparticles coming from the airway or the vasculature. Ethical and safety concerns also exist that must be carefully navigated. A number of approaches, including CRISPR-Cas9, integrase-based insertion editors, base editors, and prime editors, may be applicable to fix or insert CFTR. For example, David Liu at Harvard and Paul McRae at the University of Iowa developed an efficient prime editing approach to correct the F508del mutation. Prime editing fixed about 25% of the DNA, which restored CFTR channel function to levels comparable to those achieved with modulator treatment in the HBE assay. This provides proof of principle for an approach for a durable one-time treatment for CF caused by some CFTR mutations. Prime editing is now being further developed by the company Prime Medicine. Here, functional CFTR leads to swelling in this organoid assay after stimulation of CFTR. DNA correction of the G542X nonsense mutation led to significant CFTR functional restoration, as shown in the video on the bottom right. Since there are many CF-causing variants, scientists are also working on approaches that could help all people with CF. Here, the concept of introducing a superexon is shown. The cDNA coding for CFTR exons 2 through 27 could potentially be inserted into intron 1 of the CFTR gene. This approach would use gene editing to replace exons and introns that could be harboring mutations that cause disease. Multiple labs are now exploring this concept using a variety of editing strategies and delivery systems. This would produce a repaired CFTR gene where exons with CFTR mutations after the superexon are not used. Here you can see recent progress from the CFFT lab, where Hilary Valley and her team have demonstrated that the superexon idea has become a reality in cell-based experiments in the lab. In the red box, you can see the CFTR protein from normal cells and the complete lack of CFTR protein due to the G542X mutation. After the superexon was inserted into intron 1 of the mutated CFTR gene, protein expression was restored to at least normal levels in 11 different clones. On the graph below, chloride transport was restored to wild type or higher levels. Continued development of editing methods that can be applied to all mutations, as well as continued development of delivery nanoparticles that can accommodate these larger payloads for delivery to the lungs and other tissues, is ongoing to further advance CFTR gene insertion therapies. Finally, let's come full circle and discuss the possibility that delivery nanoparticles administered systemically might be able to overcome all those delivery barriers to allow genome editors to fix CFTR in progenitor cells to create a durable therapy. In my own lab, we have been developing sort LNPs that can target and enter the lungs from the vasculature. Here, we delivered a genome editor in lung sort LNPs to a mouse model wherein edited cells become red. We discovered that sort LNPs could mediate delivery to nearly all cells, from the top of the trachea down to the bottom of the lungs. This is shown in the 3D image on the right of a mouse lung. In comparison to the COVID-19 vaccine LNP, which is composed of 4 lipids, SORT LNPs contain an additional 5th lipid that helps the SORT LNP internalize into epithelial cells. Our team utilized our background in chemistry and engineering to improve lung targeting and address the need set out by the NIH CFF workshop. We next delivered a base editor mRNA together with a targeting single guide RNA in lung SORT LNPs to HBE cells at the air-liquid interface. This approach was able to correct 70% of the R553X nonsense mutation to the correct CFTR sequence. In the functional assay results shown here, a 54% restoration of CFTR chloride channel function was quantified, which is comparable to the activity generated by Trikafta on the other allele. The combination therapy produced functional equivalent to 85% of normal lung cells. And in collaboration with colleagues at Case Western Reserve University, who created a CF mouse model with the human R553X nonsense mutation in CFTR, we delivered base editor SORT LNPs intravenously and found that they could correct the DNA in the lungs, including in basal stem cells. Analysis of lung tissue extracted 10 days after a single injection of LNPs revealed 50% correction at the targeted DNA site in lung stem cells. If this approach can be translated to humans, the implication is that gene correction therapy would indeed be long-lasting. So finally, let's think back to the results from Intellia's clinical trial showing that liver editing is permanent. Well, we decided to track those animals with the red cells in the lungs for nearly 2 years and see what would happen. Excitingly, after only 2 intravenous administrations of gene editor lung SORT LNPs, the lungs were genome edited and stayed edited for essentially the lifespan of the mouse, likely due to editing in those important basal stem cells. Well, there's still a lot of work ahead, and I do want to emphasize the additional work, including improvements in the ability to deliver to the lungs and other tissues, manufacturing, safety evaluation, and much more that remains. But if this finding in mice can be translated to humans, it may indeed be possible to someday realize durable therapies for cystic fibrosis. So, yeah, it's great. So let's reflect on where we were and where we are going. Since the discovery of CRISPR in 2014, to the CFF NIH workshop in 2018, to more than 5 billion doses of mRNA LNP being shipped all over the world to 180 countries during the COVID-19 pandemic, and now the initiation of the first CFTR mRNA LNP clinical trials, we have made significant progress. 6 years ago, we defined goals and worked to provide answers to those key questions listed on the left. We now know that basal stem cells, ionocytes, and epithelial secretory cells are target cells in the lungs for genetic medicines. New assays and models are defining editing and protein replacement levels needed for therapeutic benefit. Emerging data is matching editing platforms to address specific mutations while paving the way for future strategies of CFTR gene addition. New synthetic carriers are available that can deliver genetic drugs to the right cells in the lungs. Manufacturing capabilities and improved regulatory efficacy and safety guidelines to support clinical trials have also significantly advanced, which you will hear more about from Joe next. We do have work to do, but the time is right to pave the clinical development path for genetic medicines. Thank you for your attention. Thank you. So at this time, I'd like to invite Dr. Joe Pilewski to the stage, and he's already here. Welcome, Joe.
Thanks so much.
Welcome, Joe. Good luck. Yeah.
I don't know about you, but I have a sense of wow That was really remarkable. I think the progress, as I've been in CF for a long time, it's incredible, and I don't know how else to articulate that. So I'd like to say thanks to Steve and JP Clancy and the meeting organizers for the opportunity to speak today about the progress we have collectively made towards moving genetic therapies into the clinic. Thanks to all of you for being here and for your dedication to improving the lives of people with CF. Hello to those of you joining online. Your courage inspires us as we gather this week to learn, share ideas, and focus on how each of us can serve you and develop new treatments. I have no financial conflicts of interest. My institution receives grant support for clinical trials. I particularly thank the many people on this list for your contributions to our progress to this talk today and for your support. Dan described the remarkable, dare I say incredible, advances in the basic and translational science of genetic therapies for CF, which we refer to as nucleic acid-based therapies, or NABTs, as summarized in the events on the upper left of this timeline. Before we review recent innovations for advancing NABTs to clinical use, let's consider the historical picture. A year after identification of the CF gene in 1989, scientists demonstrated that a normal CF gene could be transferred to cells and restore CFTR function using engineered viruses. Just a few years later, human studies demonstrated that inhaled recombinant adenovirus or adeno-associated virus or lipid DNA complexes could be delivered by inhalation to adults with CF. We learned much from this experience, notably that with careful planning, the study participants did not have significant side effects. These studies demonstrated safety. We also learned that the delivery efficiency available with technologies did not provide clinical benefit, and that more efficient delivery methods were needed. Scientists therefore re-engineered viruses to be more efficient and developed novel delivery vehicles, as Dan described beautifully. Now we move to the right end of the timeline and how we are poised to support industry sponsors and help develop the newest genetic therapies. Let's start with the path to a nucleic acid-based therapy that became possible with identification of the CF gene and determination of its expression and function. The first milestone on the path is to deliver a normal gene or edit the abnormal gene using recombinant viruses, or one of the remarkable technologies Dan presented. With the genetic material and novel delivery vehicles, the engines and cars in Dan's analogy, we proceed to laboratory studies to determine safety and efficacy in cell and animal models. Success here informs the development of clinical studies to demonstrate safety and efficacy in humans, then to study execution, and if the therapy proves safe and effective, regulatory approval. Today, we will focus on advances in the last 6 years to optimize the middle milestones in this journey, specifically innovations in laboratory models to provide evidence for safety and benefit for people with CF, trial designs and study endpoints, research infrastructure and educational tools to optimally engage people with CF for study participation. I'm only going to hit the highlights of these because the advances are too numerous to really indicate today. Since we are in the great city of Boston, let's think of one of Dan's cars as the NABT, or genetic treatment, that needs support to progress through clinical development as we visit a few historical sites from the American Revolution. First stop on our journey is Faneuil Hall, little over a mile from here, meeting place for the Sons of Liberty, who in response to British taxation of goods, hosted town halls to bring people together, just as the CF Foundation has supported transparent partnerships with basic and clinical scientists, and drug sponsors to develop laboratory models and the data necessary to demonstrate safety and efficacy. Before human studies, study sponsors and regulators must address two questions. What data indicates that a delivery vehicle will be safe or effective in people? Secondly, what is the risk to humans? In the case of NABTs, there's potential risk from the genetic material, that is the cargo, or from the delivery components, the vehicle. For example, recombinant viruses or lipid particles. There may also be risk of study procedures. Answers to these questions using laboratory models are critical, since safety is always the priority in every clinical trial. Primary cell models were instrumental for identifying effective CFTR modulators to restore CFTR function. As shown in the figure from Marcus Mall and colleagues, the change in percentage of CFTR activity in CF cells after CFTR modulator predicted improvement in FEV1 in people. In contrast, NABTs must be delivered across the cell membrane in the complex airway environment. Cell culture models may be useful, but do not fully reproduce the intact airway in the lung, so perhaps will not predict efficiency of gene delivery or correction. This highlights the need for new and novel animal models. These are important to determine lung deposition and expression, and we have a number of novel animal models like the CF ferret, or CF rat, or CF pig. These may prove useful for proof of efficacy prior to human studies. In addition, animal models are important for assessing elements of safety that typically are not a consideration with small molecules, such as the immune response, integration into DNA into the lung, or other organs. Animal models are also useful to predict safe and tolerable doses to be used for humans. The laboratory safety data is reviewed by multiple oversight committees who prioritize participant safety as they consider approving the study. The study sponsor submits efficacy and safety data and the clinical study protocol for review with a focus on risk and benefit. These include the Food and Drug Administration, 2 CF Therapeutics Development Network Committees, the Protocol Review Committee and the Clinical Research Executive Committee, the CF Data Safety Monitoring Board, and an Institutional Review Board. A study only begins at 1 or more of the CF therapeutic sites after the FDA, TDN, and IRB approve. During the study, the DSMB reviews safety endpoints to assess whether the trial is safe to continue. The site study team and study participants help to ensure safety by communicating any side effects to regulatory agencies and adhering to the protocol. Next stop on our journey is Lexington, Massachusetts, destiny of Paul Revere's famous ride, and coincidentally, home of the CFF Therapeutics Lab led by Martin Mentz, so instrumental in developing and assessing new small molecule and nucleic acid-based therapies for CF. The growing community of people opposed to British rule banded together to protect one another. Just as Paul Revere rode to protect the colonists in Lexington, our community is committed to protecting all people from complications of their CF. To do this with NABTs, innovations in trial design are necessary based on changes in the CF population. As you know, highly effective modulators are now available for over 90% of people with CF, represented in the light blue. The highest need population that is eligible for early phase NABT studies is the approximately 1,600 adults represented in dark blue who do not respond to modulators. This population is much smaller than in the 1990s when the entire CF population was potentially eligible. NABTs may one day add to the clinical benefit of CFTR modulators or replace them as a therapy. However, early phase NABT studies will focus on those with the highest, most urgent need, those who are not eligible for CFTR modulators, and an additional 5 to 10% of people who are intolerant or have limited benefit. The changing demographic informs how CF is considered by the FDA and how clinical trials are designed. Products are evaluated by the FDA based on their risk-benefit profile in the target population. Studies must demonstrate, using defined outcome measures, improvement in how patients feel, function, or survive. Regulatory flexibility and risk tolerance in the vertical direction on this graphic are proportional to disease severity, rate of progression, and therapeutic need in the horizontal direction. Extreme examples are the common cold, which would receive low regulatory flexibility, and inherited fatal neuromuscular diseases like cancer that receive high flexibility. While CFTR modulators have reduced disease severity and rate of progression, the population not on modulators has higher unmet need and will accordingly receive greater regulatory flexibility for NABTs while maintaining a focus on acceptable risk and prioritization of safety. The FDA provides added support for rare disease therapies that provide opportunities for more rapid development. Programs to expedite clinical development such as priority review, accelerated approval based on surrogate or short-term study endpoints, fewer clinical trials with combined phases or single pivotal trials. Lastly, there's support for innovative trial designs, such as the use of externally controlled trials, as we'll talk about in a minute. Discussion between the sponsor of the therapy and the regulators, the FDA in the US and the EMA, or European Medicines Agency in Europe, helps the sponsor to develop the clinical study. The FDA approach to rare diseases and the change in the CF population at highest need created an opportunity for the CF Foundation and academic investigators at the TDN to increase interactions with sponsors on specific studies and with regulators on principles that would apply to all NABTs. These interactions focus on education regarding the changing patient population and advocacy on study design and outcome measures, such as the current primary outcome measure for determining clinical benefit, FEV1. Experience from prior studies helps with the question how much change in FEV1 would be significant and clinically meaningful. Shown here is the change in FEV1 in placebo groups in orange and those treated with the early CFTR modulator, lumacaftor ivacaftor, or LUM-IVA in the dashed line, which had modest effect compared to those treated with highly effective modulators, ivacaftor and elexacaftor, tezacaftor, ivacaftor, ETI, in the solid lines. We are hopeful of providing clinical benefit as good or better than IVA or ETI. However, for design of NABT studies, we're working with the possibility that the initial NABTs may be beneficial but less effective than current best modulator therapies. Therefore, we are conservatively modeling trials to show an effect in the range of 5 to 7% predicted FEV1, as indicated in blue. If you missed the live session earlier today, please listen to the recording of Symposium 11 for a detailed discussion of trial design and a number of other issues associated with nucleic acid-based therapies. During trial design, statisticians determine how large a sample size is necessary. These estimates are required for later phase clinical trials to help ensure that there's statistical power to detect an effect from the intervention. Estimates are based on anticipated effect size and the variability of the outcome measure. In this figure, the greater the difference between treatment and control, as seen with ETI and IVA, the smaller the required sample size. While to detect a smaller effect size difference, as seen with LUMA-IVA, a larger sample size is needed to demonstrate a difference. For NABT studies, modeling developed by J.P. Clancy and Nicole Mayer-Hamblett indicates that for NABT, providing at least a 5 to 7% increase in FEV1, inclusion of 40 to 80 participants would have a high degree of statistical power and demonstrate a clinically meaningful difference in the process. This is great news for the development of NABTs because with regulatory flexibility, data from relatively small clinical studies could support approval. Now let's step back and summarize the phases of a clinical trial process and other ways to optimize trial design. The typical study progression for all new therapies is from phase 1, focused on determination of a safe dose, phase 2, focused on safety of a limited number of doses and provision of proof of efficacy to support proceeding to phase 3 studies that will really focus on efficacy with monitoring of safety in larger populations. With the study design principles for a limited pool of high-need study participants, clinical trials for NABTs will require smaller sample sizes than small molecule or modulator studies. As shown here, phase 1 and 2 modulator studies included 10 to 20 participants per dose, whereas phase 1 NABT studies employ a 3+3 design with 3 participants in groups of increasing dose to carefully assess safety. Later phase 1 and phase 2 NABT studies are likely to include 6 to 9 participants per dose, and pivotal studies will require roughly 40 to 80 participants compared to the 400 to 600 typical for phase 3 modulator studies. In typical trials beyond phase 1, a study population is randomly assigned to active treatment or placebo. In contrast, recent trials in genetic therapies have used external control data that is not collected during the current trial. This data could either serve as a component of the comparison group with hybrid trials or serve as the entire control group in a fully externally controlled trial. With the need for patient-reported outcomes on how patients feel and function during a trial, fully external control trials are not ideal, but hybrid trials would be useful. Notably, 8 of 11 approved genetic therapies used external controls. An ideal control population would be based on people receiving current best therapies with data collected consistently to ensure quality. So as is customary in CF, academic collaborators in the TDN and the CF Foundation developed the REACH study to capitalize with FDA guidance on the opportunity for creating an external control group. This study, led by Deepika Polanini and Nicole Mayer-Hamlet, is designed to establish placebo data through a prospective observational study of people with CF who are not eligible for or are intolerant of CFTR modulators. This study is particularly important because it seeks to engage people with CF who may have been late to diagnosis and under-recognized due to their race or ethnicity and may previously not have participated in clinical research. As shown in this table from a recent patient registry publication led by D.B. Sanders, characterizing the CFTR modulator ineligible population, disproportionately more modulator ineligible people are Black or Hispanic, and research participation has tended to be lower than the overall CF population. A high priority for our community is to ensure that all people with CF have access to the best possible clinical care and to research studies that may provide new treatments. The REACH study enrolled its first participant last week and is on its way to meet several goals: to collect control data prospectively, engage people with CF who are ineligible or intolerant of CFTR modulators for research using some web-based technologies, and use data for clinical development planning to support innovative trial designs. The study will be conducted at research sites with referrals from care teams to allow people with CF to help generate the important control data for regulators and multiple study sponsors to be used in the placebo or control group of clinical trials. This may allow for randomization of fewer participants to the placebo group, thereby reducing the total number of participants required for each trial and allow for more rapid assessment of NABTs so we can meet that sense of urgency. Measurements other than FEV1, so-called secondary outcome measures, may facilitate the development of NABTs by providing evidence that predict clinical benefit. The first of these secondary outcome measures focuses on detection of CFTR expression in airway cells. Assays of CFTR mRNA provide information on gene transfer to specific cell types. More helpful are assays of protein to determine cell type expression of CFTR as evidence of an efficient NABT. Methods have been established for detecting CFTR and other proteins using fluorescent labels in cultured cells, as shown here. Our need now, and with support of the CF Foundation and academic investigators, will engage to optimize methods to detect CFTR expression on airway samples obtained by bronchoscopy during research studies to provide more efficient NABT markers and identify which therapies may be successful. Another promising outcome measure is mucociliary clearance, as developed for human studies by Scott Donaldson and colleagues, with techniques like those used for the CF ferret mucus clearance studies with the RCT 2100 mRNA therapy Dan showed earlier, and as in these images from a participant before and after starting ivacaftor. Mucus clearance improvement detected by this technique correlates to improvements in FEV1 with effective therapies and may be useful as proof of efficacy and for determining NABT dose. Another approach is to measure using lung clearance index how well gas washes out from the lung, as this method detects the abnormal gas emptying that occurs in diseases like CF. Multiple studies, such as this one published by Edith Zemanek and colleagues, have shown that with effective CFTR modulators, FEV1 increases, as seen in the top panel, and lung clearance index also improves, as in the bottom panel. One limitation of LCI is that most of these studies have focused on children and younger adults who often have very good lung function. However, recent analysis of multiple studies suggests that with consideration of baseline values and severity of lung disease, LCI may provide a valuable secondary outcome measure in older participants with more disease. A final recently developed secondary outcome measure is quantitative analysis of CT scans to assess changes in lung structure with restoration of CFTR function, as shown in these images before and after ETI. A recent study of ETI by Paul McNally and colleagues in the UK and Ireland demonstrated decreases in mucus plugging, shown here, and in multiple other parameters of abnormal lung within 1 year of beginning ETI. While promising additional studies are needed to demonstrate the usefulness of CT as a shorter-term outcome measure. A C-Symposium S22 tomorrow for additional details on outcome measure and effect sizes. Our journey next takes us to Pennsylvania State Hall in Philadelphia, where leaders including Franklin and Jefferson rallied delegates to the fight for independence. Similarly, let's rally as CF care and research teams to enlist people with CF for clinical trials to develop new therapies. This effort will be supported by the last area of innovation I'll touch on today: infrastructure and tools to engage people with CF for study participation. A sign of the great progress in delivery technologies is the remarkable number of phase 1 and a few phase 2 NABT studies, including mRNA shown in purple, and antisense oligonucleotide in light blue, and gene addition studies with recombinant viruses in dark blue. The more rapidly we complete enrollment for each of these, the sooner we will learn which, if any, of the current NABT technologies will identify a safe dose and find evidence of efficacy to move forward to larger pivotal studies and regulatory approval. We must be prepared to meet the challenge of enrolling from a limited US population estimated at a few thousand adults with CF. Now please join me to hear the experience of Brady, a young man with CF who recently participated in an NABT study, and Justin, who, like hundreds of research coordinators across the TDN, serve a critical role in clinical studies.
My name is Brady Brock. I'm 25 years old and I have cystic fibrosis. My main goal in life is to get married and have a family one day. And I know that's a very simple goal, but that is my one desire in this life, I think. To have that family and to be able to be there for them, not just by being alive, but also by being healthy and being able to be the provider in the family for both my wife and my children one day. I found out about the genetic therapy trial that I'm in right now through my CF team in Jackson, Mississippi at UMMC. This is my first clinical trial that I've been a part of. The pool of individuals that can participate in these research trials is very small. I fit both categories. I have 2 nonsense mutations, and I'm also healthy enough to participate in these trials, so I felt almost a responsibility to help out in that way.
My name is Justin Anderson. I'm a clinical research coordinator at the University of Alabama at Birmingham. CF patients potentially have the opportunity to take a CFTR modulator, which has done a lot for that group of patients, but we still have patients that don't have options for that, and even some of those patients that are able to take those modulators have not responded well or have not tolerated them that well, that still need therapies. Currently, the clinical trials that are out there for genetic therapies are inhaled therapies. And so right now, it's just looking at the lung.
Hey Brady, what's up, man? How's it going? It's going well.
These genetic therapies are definitely the next step for finding the cure. Some of the main questions and concerns that patients bring to us, one most common one, I think, questions about the safety of these, and they're concerned about being some of the first patients to try this new therapy, new gene therapy or mRNA therapy. There's a lot we don't know, but I think we have enough information from other diseases and using these kinds of therapies to kind of give them some idea of what could potentially happen.
With these trials, we really don't know the long-term effects, but I do know the long-term effects of cystic fibrosis, right? So the possible benefits kind of outweigh the possible negative risks associated with the study. When it came time to actually take the drug that is part of the study, there was a little bit of nervousness there. I think the team has been outstanding in addressing all of my concerns throughout the process.
It really is a team effort of partnership with the patient, with their clinical team, and then with the research team. What makes me most excited about the genetic therapies is seeing the reaction from patients that haven't been able to be any part of any of the modulator trials because they've had mutations that they're not eligible for. Just seeing how excited they are, I think makes it worth all of the work that these trials come with.
What I would say to people contributing to cystic fibrosis research is twofold. One, of course, is thank you so much. It is easy to feel forgotten in this post-Trikafta age, but seeing your work makes me know that that's just not the reality. And the other side of that coin would just be to keep it up. Keep going. You were blessed with the knowledge to do this work and blessed with the ambition and the drive to continue doing it even when it gets hard. So I would just encourage you to keep up the good fight.
Wow, what commitment. I'd like to thank Brady for sharing his story and committing his time and energy to clinical research, and to both Brady and Justin for being such great role models for study participants and research coordinators. To lay the groundwork for genetic therapies and complete clinical trials, many resources have been developed for each of the key stakeholders. We have discussed some of the recent efforts with the FDA. Some of the other existing resources are shown here. For our last few minutes, let's review a few of the powerful new initiatives to engage care and research teams and the CF community. Our discussion today is on opportunities to develop nucleic acid-based therapies, but we remain laser-focused on symptomatic therapies targeting infection and improving mucus clearance in people with CF who are not eligible or intolerant of modulators, improving access to clinical trials for all people with CF, especially those from historically underserved racial and ethnic groups, To meet the need for research engagement across our entire community, the TDN began a regional cooperatives initiative in 2023. To do this, the country was divided into 20 regional groups to help establish relationships between CF care and research teams, something that previously often didn't happen, with the goals of building research knowledge, improving access to clinical trials, and increasing referral opportunities. Each of the care center team members can participate in virtual meetings with one or more regional cooperatives and contribute to our research mission. To further support the care and research teams, the most recent innovation, the Research Connection Hub, will facilitate information sharing on all aspects of clinical research. The hub went live this week thanks to the vision and incredible work by Jill Van Dolfsen, Trenton Gordon, and colleagues at the TDN and the CFF. The hub is a searchable website that provides information on actively enrolling studies, new initiatives, and key study updates, education on how to discuss clinical research and study referrals, training and tools for care providers and research personnel, and handouts for people with CF and their families. Care and research teams can log in to mycff.org to access this remarkable repository of resources and clinical research information. For our CF community, in addition to resources on cff.org, how else can they get involved? First, it's critically important to discuss research with your care providers. Second, contact the clinical trial navigator who can explain the clinical trial process, answer questions about participation, provide reports of enrolling trials, and facilitate contact with a research site. Katie can be contacted by this email, or people can fill out an online form to get started. To summarize, today Dan and I have reviewed the path for our journey to genetic therapies for CF, highlighting some of four innovations that provide support for our journey and make me optimistic, hopeful, as Cam said earlier, that we will succeed. First, remarkable technologies to improve delivery of genetic material and to develop gene editing approaches for a long-term cure. Second, laboratory models to address safety and determine efficacy signals predictive of clinical benefit. Third, innovative trial designs and study endpoints to conduct efficient trials and conduct them with a sense of urgency. Finally, infrastructure, educational tools, and technologies to engage people with CF for study participation. Okay, so we've visited a few sites. Now we're going to stop lastly at Independence Hall, where we remember our freedoms and for inspiration to continue our work for people with CF and their quest for health. As we think about our mission, please remember that all of us can contribute to the development of new therapies. Clinicians and clinical care team members, please participate in virtual regional cooperative meetings and partner with the TDN centers. Use the Research Connection Hub, encourage participation in clinical trials, and make referrals. Basic and translational investigators can innovate in the laboratory and clinics on preclinical models and better nucleic acid delivery methods. Research coordinators and staff improve education on research and recruit participants for trials. Most importantly, people with CF, like Brady, ask questions, learn about clinical trials, and seek study participation. Thank you for attending today and for this opportunity. Please join our research efforts and keep up the fight for people with CF to live long and happy lives.